Well, good afternoon. It's good to see the stragglers here, the diehards. It's an amazing conference, Stuart. If you can get people coming for workshops on Monday and they're still here on Saturday afternoon, that's pretty darn good. No, you guys are super. And I was actually speaking to one of the itinerant speakers, who I hope will convert to a regular speaker earlier today, and he was saying that it's a really great community. People who care about this topic and are coming and talking about it and thinking and working hard. It's so again a tribute to Stuart and to you for coming and thinking about this, and we'll work our collective consciousness. So this afternoon, let's talk about where did it all come from. So our session is entitled The Origin and the Evolution of Life and Consciousness. And we're going to start with Dr. Bruce Damer of UC Santa Cruz on the origin of life and consciousness. Dr. D.
Thank you. I want to thank the two Stews, Stuart Kaufman and Stuart Hameroff. Stuart Kaufman had recommended me last year, and he was doing more of an origin of life theoretical biology approach, and that's how I got put together with you, Stu. And thank you very much for this kind offer. What you're going to see today is actually the first time this work from science has been presented outside of our community. So this is the first time I'm presenting this in an audience where it isn't two-thirds chemists. So this is quite a little bit historic. I was writing to our team last night saying, this is it. This is where we launched this thing. So thank you again. So what I'm going to do also, to provide you some grounding, I'm going to pass around an artifact, spend a few seconds with it. What this is stromatolite, some of the oldest evidence for life on Earth. And I'll be showing you another during the talk, so feel free in the next 30 minutes to make a connection with it. So off we go. So this is a novel approach to the origin of life. This is the first end-to-end scientific theory, hypothesis, testable in the lab for the origin of life from basic molecules to cellular life. It doesn't go beyond that, of course, but it gets you to that point. And it possibly has implications for consciousness and everything else. The disclaimer, this wording was assistance from someone here at the meeting helped me with the wording so I could make it more fitting with what you guys are working on. So try to treat this abiogenesis stochastic cycling implication seriously, and you'll see the cycle because it's supported by empirical evidence. We found an engine that can boot stuff up in chemicals, but where it meets the hard problem, the Chalmers hard problem, please treat this as playful speculation. So what you're about to hold in your hand and what I'm holding in my hand here is new for science. This is some of the oldest evidence for life on Earth, 3.48 billion years old, that was discovered in the Pilbara last summer field season. And these little red nodules are stromatolites. They're layers laid down by organisms which sequester sand grains together. And then they stack those and they grow up on top so they can keep access to the sun. That's the whole name of the game. And they built these rock textures or fabrics, all cones and domes and you name it, and flat pieces. And this is the sole story of life on Earth for just about 90% of the time. And this is still the bulk of the story of life on Earth. We're this rarefied thing called complex animal life that popped out maybe for about 2% of Earth's history and will go away. But microbial life, microbial community is the story of life on Earth pretty much and by bulk. So I'm going to take up not your hard question, but 3 other hard questions that might be related. How did the non-living world, how did non-living matter atomate itself into the living world? That's one. Why did life bother to come into existence, or did it have a choice? There's your free will for you. And this one, which is very speculative, and I'll be quite happy to get critiqued by this group. What is the source of all, an arbiter of all experience? Which somewhere in there is consciousness. So a little bit about me. I have two career paths. One is working with NASA on spacecraft. So there's me at NASA. I helped NASA create a whole new architectures for missions to the outer solar system, but also to the asteroid belt, if you will. This is my design for a human mission to an asteroid. It's the first reference design. I've done about 25 projects for them. This is asteroid capture in a balloon structure and manipulation with gas called Shepherd, which can also make biospheres and do mining and get fuels. And this single invention by a team of 3 of us has the potential to open the solar system. So this is our current initiative, is to find a government program or government private to start running demonstrator flights and try to prove this technology to allow human beings to extend the biosphere into the solar system. So that's one aspect of my work. We're now working on site selection for the next rover. This is the Mars 2020 rover. It comes down on a sky crane. Its primary mission is search for life. It's the first NASA mission to search for life on another world, not just do the geology primarily or find the water. And we're arguing with NASA, we're debating, we're trying to convince them to go back to this location. This is a picture taken by the Spirit rover about 8 years ago of part of a rock outcrop, which was a hot spring that could preserve life just like this, about the same age. So we have a shot at finding a life on another world if we go back to Columbia Hills, which is where Spirit is sitting there right now. So rolling back the clock, how did I get interested in origin of life? I was raised in Canada, in Kamloops, British Columbia, Canada, and I love to walk in the nature. And I love sagebrush because to me it was like little forests. Like you're flying above forests when you walk through sagebrush. They're like miniature oak trees. And I was walking out in the spring of 1976, and I bent down, and there was this beautiful Mariposa lily coming up through the ground that had just been frozen. And my little 14-year-old brain just flipped and said, how did this structure, how did this beauty emerge? This is a beautiful plant, it's beautiful, and there's a bulb or a seed somewhere down under the soil that has instructions to do this. And then I stood up and I realized all the plants are joining some kind of instructions. And then my brain sort of went into hyperwarp speed, and I went back and said, well, where did it all start? Did it start with one seed or one instruction? And then I thought, hey, I'm a nerdy kid. I'm a nerd kid in a town with no computers, but I'm a nerd kid, and this would be a great problem to work on my whole life. So that's when I committed to working on this problem. And I learned about this fellow. I was reading about Albert Einstein, that at 16, he had thought experiments. He actually had them even earlier, but he had one where he's running alongside a beam of light, and he sort of sees in his imagination, it's sort of a delivered vision this guy got at 16 about the compressing of the waves of light and things that led to special relativity. So I was on the verge of thinking I could do that when this happened. And I drew this out. Like in my mind's eye, right in front of me, a thought experiment, a Gedanken experiment started happening. And it was a bundle of molecules. And I didn't even know what molecules were. I knew they were kind of things. I'd read some chemistry articles. And they're moving. And I'm formulating a question. You can ask thought experiments questions. I'll ask this one. How did simple molecules self-assemble into things that could do things on their own? And instead of getting that question out, it asked me the question, said, figure out how we made a copy of ourselves. Figure out how we made a copy of ourselves. And my little brain flipped and said, well, if you have a TR7 automobile, it's made in a TR7 automobile factory, which is a bigger machine, and I don't see a bigger machine here, so it's impossible. And the little bundle of molecules winked. And that's what I've been working on for 30 years. So here I am 40 years later on the trail of it. So this is me at Bumpus Hill, which is a hydrothermal field up in Mount Lassen National Park. It's actually a very dangerous place to be standing because Mr. Bumpus lost his leg there 150 years ago. What I'm doing is putting a slide tray into a fumarole vent, which has gases coming out. This is the slide tray. Testing the theory that just vent gases alone with humidity could allow us to polymerize an important biopolymer. And it worked. This is the first time it's been done in the field. And this is our gel. And if you see that little blob there, that's RNA. We made RNA without enzymes in a fumarole vent environment. Because our lab goes to these environments and tries to do it in the messy chemistry of nature, for goodness sakes. This is what we use in the lab to do that. It's a device that cycles around 24 dishes between hydration and dehydration stations. And we have a 90 Celsius atmosphere. There's our hydration station, our dehydration station. And we put in reagents that are the building blocks of life, basically. And we can make polymers. We can make the building blocks of life without using life. And this is the first clue to how to do this. Here's our evidence, because our colleagues are very skeptical. They're chemists. They're incredibly hardcore materialist people. So you have to really do this thing right. So here's the gel electrophoresis, and those black bands on the right are our product. We invented a technology called nanopore sequencing, and we ran it through our own device, and it counts the base pairs as they're going through a pore and gives you little blockades, and you can actually do digital, like analog nature to digital directly. It's a really cool technology, and it worked there. And we blasted it with X-rays to see the stacking order. And then we discovered that we could get these polymers, these biopolymers, inside vesicles, inside lipid. The membranes that actually float in from space, actually through dust, they come in and they land on the early Earth, and you got your membrane supply, you got your amino acid supply, and some of your building blocks are coming in from space. It's kind of a panspermia, but it's not a fully formed organism, it's just a building blocks. But we proved now, it's pretty much taken in the field, that we can grow polymers this way through basically drying things down and letting the water go away, and you can make these things. It's like nature's way of making biopolymers before life took over. And they can become encapsulated in these containers. And then you can subject them to selection by their trillions. Now let's take a look at how this works. So that's all the chemistry I'll show you. Let's go to a metaphor everyone in this room understands, computers. So if you're like me in the mid-'70s, your first computer might have been this clunking thing with switches on the front with a paper tape input. It wasn't quite that bad for me, but what you had to do is punch the program into paper tape. This is how Microsoft started, which was the basic the loader to boot load, and then to load the basic language to then run real kind of language. Well, what if you started with a random puncher, but you had an infinite spool that made just zillions and zillions of program tapes, just trillions of them. And they went into a reader, and the reader went into your primitive computer. This is my Altair 8800. And it has an energy source, and it cycles. It'll try to run those programs. And there's this little processor, and there's this little instruction. And they either crash the system, it goes into the crash trash, or it runs, play again. And you get program A. Program A can go back into the random feeder and get added to with, let's try B with that, and C with that, and D with that. Let's just try to extend that. Well, over time, you'll develop more functions. You might evolve a computer that will now have a screen and a keyboard if you're evolving both together. Keep going and you might get to a laptop. Keep going and you might get to your iPhone. So this is a model for evolving software and hardware together. The only thing different is that you're doing it randomly, so it takes millions of years. So we hire a cheap form of biology called engineers to do a slightly better job than the random puncher. And we get our programs written and things like that. There's no engineers in the audience, right? So this is an inefficient way of booting up a system. But this was perhaps the only way that nature had available to it, but it works. This method works. So let's replace this system. Let's go out into the wild and try to find in the world where we could do this. How could nature do this? Well, it's quite simple. The polymerizer is membranes and organics all smushing together, coming in, concentrating in pools, and polymerizing. That's what I was doing in Bumpass Hell. I was just doing that. So it makes polymers, which are your tapes, your punch paper tapes. those are fed into a computer called a warm little cycling pool. There's our Charles Darwin, who in 1871 said, I think life started in some warm little pond somewhere. And he was extremely prescient. This one little paragraph is extremely right on. Absolutely. So as a source of energy, it's pumped by a geyser system. So what are your programs? They're called protocells. They're random collections of polymers in random little bags of things called lipids, lipid membranes. And how does your processor work? It's Charlie again, natural selection, a form of early natural selection where he says, oh, that one dislocates and blows up, and this one's fine. This one's more robust because the polymer did something to stabilize. And it kind of like microtubules or cellular structure might stabilize the membrane. Oh, that one can go forward and go again and again and again. Same system. So we put it all together, and this is artwork done for Scientific American, which will be in an article in August issue to announce this to the general public. Hot spring origin of life. So here's the big picture. You've got your landscape 4.3 billion years ago, called the Hadean period. You had your volcanoes, your Fred Flintstone volcanoes, and your dirty atmosphere, and your chocolate-covered oceans, or colored oceans, and it's all infused with iron. That's why this stone that you're handing around has worked its way around is so heavy. It's full of oxidized iron. So we synthesize in the solar system all our building blocks, as I was telling you before. We accumulate them on land in little pools, get our organics, until they get concentrated enough to do chemistry. Because if they go in the ocean, they diffuse. So that's why the field's turned away from an ocean origin now. Just can't do it. You can't get chemistry to work. That's very, very difficult in a diffusing, pregorgine type environment. So then your organics are floating along. They're in their pools, and you're forming membranes and all kinds of goodies. And they happen to flow down into this optimal pool that's getting filled and draining and filled and draining, or filled and drying and filled and drying. And that creates a pump, a natural pump, to pump these polymers in through their film phase. This is where they're being written like paper tapes. That's where they're being tested by Charlie Darwin's idea. And then they accumulate in the bottom in this gel. That was our big insight. This is our big insight because this gel ends up becoming the common ancestor. It's a new idea for science. So once this gel it's just a smush of bubbles that have learned how to adapt and grow. And once they're tough enough, they can get washed downstream. They can go to other places. They get blown by wind if they're dry. And they start evolving and adapting. Then they start sharing early genetic functions, and you get the roots of the tree of life. So we found the roots of the tree of life, which is a cool thing. And they start adapting to tough environments like the seashore. Lacustrine environments, this is estuarine environments, and salt just blows you up. If you eat a whole bunch of salty potato chips, you gotta go drink water, right? So we have to learn how to get rid of salt. That's why another reason you can't have an origin in the ocean. You need advanced technology to deal with salt. And once they get to the seashore, they've got all this sand washing over them, so they have to cement those sand grains together, and they create stromatolites at lake shores and at oceans. This is a model for the origin of this that was so dominant in Earth's history and will dominate Earth after we're gone. So here's a little view from below. So here's our little hydrothermal pool. Here's our cycling system. And really, it's all about the cycling. You have to turn the wheel and go through transitions to do this kind of a trick. It's all about the cycling. And you eventually get the progenote, the boot up phase of life, which is now becoming a field because we have a model to make these in the lab now. We can grow these in the lab, and we have sort of a challenge to different teams. You can grow these progenotes, and they can start booting up the functions of biology. Caveat. This is proto-natural selection for people who study this, and it's determined by physics. It's thermodynamic efficiency and statistical mechanics running, and what it does is pushing entropy out of the system and pulling syntropy into it. And it's doing, for those of you who study this kind of things, this is Stuart Kaufman ideas, it's doing stochastic hill climbing through noisy space till it gets to the adjacent possible. That's a Stuism, right? So, but biology comes into this system which is acting like biology We got it for free, and it says, oh, I'll replace that with this. Oh, we found a function to make a pore. We found a function to do polymerization without drying down. We'll replace that. We found a way to make peptides and proteins and more. And so basically, it's booting up an OS. This system is capable of booting an OS. And eventually, this keeps running long enough, you get this. And what happens when you get a beautiful rainfall in California, it's been dry for a while, you get an explosion because you have a three-phase system. You have the dry, dry, dry. What's happening in dry? When systems are drying, the plants are getting ready to survive the dry season. So they put all their important polymers inside little capsules called seeds that aren't subject to hydrolysis, and then they lie around and they get blown by wind, they get washed by water. A rainfall event comes, it's a violent event, it distributes things, and then the moist period is when they start their metabolic process. They start sharing, they start generating. And then they get ready to make those polymers again in their punch paper tapes. It's the same thing. So we found a natural setting that is still patterned like a hand and glove onto the world that's around us. It's still patterned on our bodies. We grow and die based on this pattern. This is what made biology, at least purportedly. So 3 hard questions. How did non-living matter itself animate itself into the living world? Cycling. No need for a crater, no need for consciousness or any other action other than the physics of the universe. Why did life bother to come into existence, or did it have a choice? It just emerged given the right conditions. So in our quest now to try to find life on Mars, I did a presentation for the JPL and headquarters and big group in Pasadena. I had to do the first talk on the origin of life on another world, as in 12 minutes of terror, just like when the vehicle's coming down, they call it 12 minutes of terror. This is my 12 minutes of terror. 300 people in the room, mostly geologists again. And literally, hot springs, life emerges in this model. And unfortunately, for that life, if it did, Mars is dying. So Mars is warm and wet, and then it starts to die. And about 500 million years in, horrible things are happening to Mars. It doesn't have a magnetic dynamo in its core, so it doesn't have protection of a magnetic field, and there's an immense amount of atmosphere being blown away. And by 1 billion years in, the oceans are starting to dry up and sublimate and disappear. So if life is on Mars, it's like, ooh, this is not a very good place. I have to go for refuge, and that means through the pipes of the hydrothermal field, underground to the salty, wet rocks, and then they're stuck. If there's life there, it's stuck. They can't evolve very much forward. They're stuck in stasis until the sun, goes through a red giant and destroys the solar system, or the inner solar system. So it could just happen everywhere on these rocky worlds that have exposed land masses, enough feedstock, and enough cycling that came from the cosmos. It just could be happening. But it's extremely rare to have complex life. Totally, incredibly, impossibly rare to have complex life. But you've got trillions of exoplanets. The thing is that in order for us to get complex life, these guys had to, the reason they're red is because they're generating oxygen and they're fixing the oxygen in a process called rusting, and they're dropping the oxygen down to take it out of the atmosphere and the oceans and the water bodies. The iron is basically getting sequestered, because if they can't do that, then they can't put free oxygen into the atmosphere. So they have to fill the buffer first, and that takes 2 to 3 billion years to get an oxygenated atmosphere. And anything can happen to a planet on that time scale. Big impactor resets it, it dries down, et cetera, et cetera. We were fortunate. We went through snowball Earths. We went through, and we got to oxygenation. Then we got to complex cells. To get to us is extraordinarily rare even after that. So the hard questions, this is the one that's maybe pertinent to this group. What is the source and arbiter of all experience? And here's where I'm going to leap off from this platform. But it's an interesting platform, because if you unwind life to its boot up sequence, you can learn about what runs the living world. And what we've discovered is 3 properties that might have a bearing on what you're working on. And if we can discover those boot up properties, we may discover what consciousness is within the booted up OS. So I had a dream one night, and I dreamt I said, wow, we're like, at the moment now, we're at the boundary between physics and biology. And for years I've been doing a dance with, I call her, Madre Nature, and saying, I'm getting close. 10 years ago I said, I'm getting close, and I had my box, which was my giant computational simulation engine I'd built. And she just laughed, a belly laugh. I will not divulge my secret to you without your puny box. So I switched to chemistry, because the computer is just woefully inadequate to do this kind of work, so we had to go to chemistry. Now we're getting really close to this. We're getting to the point where we may be synthesizing DNA through self-assembly. We're getting real close, we think. So Mother Nature, so I was back there, she's got her, her frock on, and she's saying, I'm making the cookies, but you're not going to get any, that kind of thing. So this is where I'm doing all my thought experiments, at this boundary. So one night I had a dream, and I saw the pure plane of physics, moving, moving, moving. And then suddenly in the plane opens this divot, kind of like if you're doing golf and you open a divot, a sort of a pocket. And I looked closely, I said, what is going on? Why is it pushing physics back? And I look in there, and there's something going on, and it's the usual protocell, but something going through the membrane, little molecule there, hitting something there, which decides to do an action. Boop, 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 one of Stu's autocatalytic sets. And I realized if this was not in the membrane, and this didn't pass through in a way, it would never happen. So the story of this part of the dream was biology takes improbable events and does this magic trick to it. It takes them and makes them more probable, more probable, more probable than it makes them actual. Kind of like quantum mechanics is supposed to do. Don't ask me, I know nothing about quantum mechanics. So it's got this property of probability tweaking, and it's a massive scale. Now physics has the same property, but it takes a long time. It's a cumbersome process. So just after the formation of the first stars, you're not going to see gold atoms anywhere in the universe. They're just impossible because the building blocks aren't there. But after, a few billion years, gold atoms can emerge because there's heavier building blocks been made by stars. So it does the probability thing too, but it's just really cumbersome. But when life starts, all kinds of things happen. So then I was shown in the vision 2 protocells next to each other, and there was a dip in between them. And the dip was deeper than I thought it would be. And the dream said, watch how this works. And there was stuff flowing back and forth between the dip. It said the connections are deeper than we could suppose in the protobiology, in the way to biology. The connections are deep. Then it said, are you sitting down? Because these dreams always say, are you sitting down? I always say, I'm lying down, because it's going to scale up. So it scaled up, and it started scaling up to more and more protocells. And like, whoa, this is a huge system. Just this alone. This is now doing more improbable things, boom, 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 and it's talking about them. Hey, I just did this improbable thing over here. Oh, I did one over here. I'll share it with you. Improbable thing, boom, boom, boom. This is a powerful system. This is the booting up of the progenote world. We're missing a piece, though. So as it scaled up, these Cartesian plots occurred hanging in space, and I saw these curves. Probability, engine going up, connection going up, and then there was this third curve. It was like, what's the third curve? And then it did a funny thing. The dream did a funny thing. It shook the system, and I saw our good old friend, informational polymer, appear, and this like light poured out of the system, and it reflected back. Oh, that's pattern. It's invented patterns called information. It's like a patternome, and that's the third element. So then my little brain started rolling its dice, and I realized these are deeply connected. There's a probability engine operating, which can't be done in physics. There's a connectome operating, not a neural connectome, but just chemical messaging that can't run in physics, and a patternome, which is the first little oligomers of genetic sequence. And it's like these systems talk to each other rapidly and tightly, and they scale super fast, and they create something super huge. And so even in the Progenean, even at 4.3 billion years ago, when you have sludges everywhere, they're barely alive, and they can't even divide themselves. This is a massive, powerful computational, expressional probability tweaking machine before even the formal transmission to life. So what I would put to you or offer to you is that these properties are all you need to explain all phenomena. These 3 interacting properties, and they generate all observable phenomena. They might create all experience if they're like apps, running in this OS called life. And human consciousness, primate consciousness, which is very specific, different from earthworm consciousness, samples only a small fragment of this. And we sample it by holding a mirror up to another piece of broken glass, to another piece of broken glass, and we're able to sort of see something. But it's all different for all different people. An autistic person would see a different way, and all people culturally see different way. An Aboriginal person would see it a different way, feel it a different way. And it's all different, it's all relative. So the message of the dream, so usually what happens in these dream experiences, at the end I get my shirt collar grabbed, like, you listen and you listen good. I try to get this message through. And the message was, you primates, you monkeys, are only at the very beginning of sensing and studying this field. You're just starting. That you should be wary of naming it and modeling it prematurely. It's huge. Because if you come to a molehill and say mountains, we can characterize them, and then there's a hills there, and then there's Himalayas there. You're going to never go to the mountains, right? You're going to stick with this old molehill model of everything. The one that came into my heart and mind at that point was, wait a minute, maybe we are the exquisite instruments into this field, human beings. Maybe the human being is the greatest instrument ever created by this very process to sense this field. So the people who are working on all the technical parts of the science are using their minds to go into the field, and they're using science. But the people who have deep and profound and intimate connections with the world or each other, or something we haven't even figured out, are also exquisite instruments of science or of understanding. We are the best instruments we'll ever make. Human beings, humans' bodies, minds, and hearts are the best instruments, potentially, to work on this thing. So with our cells and our tools, we can explore this field. So in a sense, the dream was inviting us to keep exploring the field. Don't name it so much. Be open to what it teaches you. Learn to speak a common language amongst ourselves so that we don't get all tripped up. And that one day we may come to a more full knowing of it. But this is a process. This is part of our evolution. But we're exquisite instruments to do this job. So I want to leave you with this. Everybody presents models that maybe can be possibly worked on, and maybe you guys can work on. They call this rotary roadmap of consciousness or to consciousness. So in this model that I presented to you, the universe is a cycling system. It has to cycle, it has to turn. Stars are born, they explode, they reform, they explode. But it tends to be two phases, like a two-phase motor, like a lawnmower can mow the lawn, but not like a three-phase motor that can do, three-cycle motors. So it doesn't have memory, right? It doesn't have a memory. So in our work with our colleagues in physics, we're literally coming to the point where information in physics is more binary. It's not linear, it's not a memory. But we are now, through self-assembling polymers and testing them, we're finding memory in physics, but it steps into toward biology, found that boundary. So you're going from two phases to three. But that system works for billions of years such that the three-phase cycle can emerge, which has memory. It has bodies. It has an intermediate phase, the progenote, the little sludge. Intermediate phase, it can pick things up, it can learn, do all sorts of things, learn chemically or innovate. And that animates matter into living systems. Then you get to neural dynamics. And that's a big transition, perhaps. That's the fourth phase in this cycling system that's using all the previous phases. It's like stacked upon stacked upon stacked. And what that means is you can have learning in a system. You can have goal-directed behavior. That's a new technology. A paramecium can do it ostensibly with just chemical circuits. But when we get nervous systems, we can do it much better. But that's a new phase. How about consciousness? Is this a fifth phase that the whole thing goes through? Dick, 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 dick. Is there something there? Because we know those previous phases work. We can characterize them. We can study them. That fifth phase, what is it? What is it? What is it? That's your department, perhaps. So I want to thank you for your attention. And these are our collaborators on the Origin of Life work. And if you have Scientific American, you'll see our publication in July or August, which will introduce it to the general public. Thank you very much.
Fantastic. That was worth waiting for Saturday afternoon, for sure. Thank you, Dr. Damer. So are you enunciating an imperative for the human species to travel beyond our Earth?
Well, I think I dig it.
Yeah.
I dig it because I
Well,
think,
you work for NASA, right? Come on.
Yeah. I like it because it gives us immense perspective.
Yes.
So for instance, if we're able to, it takes the pressure off the biosphere too, because a living system is just trying to bust out, and we're potentially the reproductive organ. So Gaia is saying, yeah, take it all, as long as you do the act and the messy act of cell division, right? So cell division's messy. Birth, as all women will know here, it's a messy process, but it's good because you have another. And so if we are the mechanism for Gaia to go forward, because in 100 to 200 million years, the sun's on a heat curve, and the terminator's approaching, right? This is in Lovelock's last book. Venus was gobbled up by heat death. I mean, atmosphere left and went in the atmosphere, and horrible, it's 900 degrees on the surface of Venus. That's coming to us. And Lovelock's calculations are it's 100 million years out. So when that happens, we go to runaway greenhouse, no matter how much or how little CO2 we have in the atmosphere. So it's over for plants on land, it's over for animals. Eventually the oceans will be gone. Microbial life will continue all the way to the heat death of our planet. But we're at that 1%, 1 to 2% of complex life that, say, a planet like this can sustain. We're coming to the very end of it. So if Gaia exists as some kind of a force of nature, she's like, we got to do this thing. And what that means is getting life out of the gravity well and creating other biospheres. And so Shepard, the design of Shepard, can do that because it can encapsulate a large asteroid, melt it down to a rock. We can introduce biology, and we can feed people in space and construct stuff in space. It's a sailing ship invention for space.
It's our imperative to survive. A question here. We're going to take this one first.
What can you say about the implications of your work for the Fermi paradox?
Now that's the thing that they should be out there, but we don't hear from them.
Yes, that's right. Yeah.
We're all little green men. I think I did a talk at the SETI Institute where I added 4 new terms to the Drake equation, which was the to boldly go terms for Star Trek, and Seth Shostak was there, so we had a good laugh. But it's so extraordinarily rare. Technological civilizations have done what we've done. It's just so rare. So I wouldn't be surprised if they're separated in time and they're separated in space, and just never contact them.
Next one.
The reason why this is relevant is because if we make life more likely to happen, to emerge complex life, then we have to account for why we don't see more complex civilizations in the universe. So how much of a warning is this for us to pay attention to that issue?
I think what this is, as we look back and with the best science we can do, and we find out we're so special, not in church ladies special, right? But we are incredibly special. We're incredibly precious. The earth is precious. We're precious. We really have to take care of this place and each other, and just like, wow, look at, we're here. And and we have a responsibility because of all the effort. I sometimes have conversations with these guys, and when I'm doing mental activity at the progenote level, I say to the smudgy progenotes that are booting up to life, I say, guys, it's going to work, we think. Mars, it didn't work out. We're working a little bit of overtime. I don't tell them about all the stupidity that humans do. But I say, work the overtime, clean our atmosphere, become life, clean our atmosphere, do all the things, because this one might pan out. And they go, yeah, right. But in a sense, this is your ancestor. This is your ancestor. And we owe it to this system, having persisted, whether through will or just through just doing it, just cycling, to be, in a sense, honor the incredible labors that this ancestor did, so we could be sitting in a room breathing oxygen and eating the ancestors for lunch, and making technology, and then asking, and then looking back at them, finding them. It's an amazing moment.
Next question.
This is a fascinating talk, but for any of this elementary algorithm or whatever you call it to work, you need two things. One, you need energy, which is okay, we have them. But what's really needed is a mechanism for the entropy to decrease consistently. A consistent way for your entropy to decrease. Where is that mechanism?
Through wet drying, wet-dry cycling. So
The wet, dry cycle.
wet-dry cycles.
How would the entropy be decreased?
So you're going, what happens? You have these films floating in the water, and you have building blocks, monomers. The system dries down, goes from a three-dimensional medium to two-dimensional, and then evaporation occurs, taking all that entropy out. And then between the films, the building blocks line up, and then water leaves, and they go click, 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 and form a structured polymer, which nature does with ATP. Nature kicks water out with ATP. And so that's where your system undergoes that transition.
Positive feedback?
Sorry?
Some positive feedback mechanism?
Yeah, and then it cycles again.
Okay.
It keeps going and going and going. And that's why we're elongating and growing more polymers away from equilibrium.
So I've read that part that you just explained. Peptide bonds don't form well in aqueous solutions. They get destroyed by hydrolysis. So is that how you're saying that works?
Yes.
Yeah, okay.
Exactly, yeah.
Oh, yeah, go ahead.
And a related question, Jerry Pollack's work with fourth phase of water and ordering, he talks about the exclusion zone being set up by optical pumping. Would that play a role also in the entropy reduction, perhaps, that Dr. Chua was just asking about?
It's possible. We don't need it at the moment. We're dealing, it's possible. And quantum effects could be possible in this epoch too, because ultraviolet light, we think that the first pigments were polycyclic compounds, and that would involve some kind of quantum effect to turn that into an energy source, because this early life can't stop just being dependent on the fast food outlet of the hydrothermal field. It eventually has to make its own food. And that's a huge transition for the progenote world to do photosynthesis. And it's not going to be with chloroplasts, it's going to be with something much simpler. And that's where some of the quantum effects might come in.
Quick question, two last quick questions.
Okay. First one is just a comment. If you're curious about the Fermi paradox, Google "They Are Made Out of Meat." It's a great little movie.
Okay.
Yeah. Because they're not here because we're made out of meat, and that's what intelligence is not made out of anymore. It's all made out of metal and silicon. Anyway, I've had this argument with several creationists who make the point that if you take the time at which the sun first started to fuse, and the time at which we have evidence of life in the rocks, it's a very short time. It's a surprisingly short time. And they make the argument that it's too short. But I've read a couple of papers that say that it's quite possible that protogenotes may in fact evolve in the interstices in rocks, essentially asteroids. Have you encountered that argument? And if you have, what do you think of it?
The chemistry only takes you so far, so the building blocks are going to be forming in the solar system accretion disk, especially as the sun heats it. And then they come in the atmosphere. Now here's a fun one for you guys. We can now do the numbers on this thing because of what the comment was back here about peptides breaking down in water. So we know the outer bounds of which we make a nice peptide, it gets encapsulated, it cycles back. It's got to do it in probably hours to days. There's no refrigerator for us to put our stuff in. So we have outside bounds. We have a box. Like progenotes have to cycle at X rate, and it's hours to days. It might be minutes, maybe 23 minutes like Old Faithful, or 73 minutes filling, and et cetera, or through dew cycling, through day-night cycling. It's fast. So we can get to progenotes, potentially something that starts to evolve and adapt and then fails many times and in many places on ancient Earth. And so there can be starts and failures, starts and failures, starts and failures, starts and failure. And how we get over that, the big gap, how we cross those huge evolutionary gaps is that something started inventing something cool, but it's dying because of all the waste product buildup. Wind comes and blows a piece of it to the next pool and shares that innovation with something else that has something else. And we have horizontal gene transfer, the roots of the tree of life. So that system can stay ahead of its ricketiness. You know, and a huge asteroid hits the Earth, washes an entire island over with a 1,000-foot tsunami. That's fine. The cataclysm will wash the system out and allow the few remaining protocells to start again. So this is why I don't believe in panspermia, because, you know, life is so hand in glove with this planet. It grew up with it. It changed the planet. It created new minerals, for goodness sakes. We create minerals. You know, our cell phones are phonite. You know, our brain case is a mineral that contains a liquid crystal mineral called the neural structure. And one last point for you guys. So I'm standing around with geologists, and I said, okay, let's do a thought experiment. We'll build a time portal. You can walk through it and go to 4.3 billion years with your spacesuit and your environmental suit and everything. And we'll go to these pools. And we go to the pools, and we see the sludge in the bottom of the pools. The geologists would say, that's just sort of a mineral fusion sludge. The biologist said, that's kind of interesting because it's kind of growing. So we come back 100 years later, and the sludges are in all the pools, and some of them have turned black. And the geologist said, minerals don't turn black on their own. Well, and the biologist, let's pipette that thing, put it in our sequencer. Oh, there's a little short oligo of some RNA-like thing or DNA-like thing, and there's a lot of them in there, and they're making black pigment to shield the whole community collectively and introduce photosynthesis. So this is where geology and biology separate. Geology used a hard minerals to make their forms. Biology used soft minerals that could learn how to make their own solutes and keep growing and building. And here's the last piece for you guys. It's sort of an extension of the talk. Here's the beautiful thing for philosophy from this. We didn't start from competing protocells. We started from a collaborative community unit. It's a network like the internet, where every part is contributing to the whole. This could be proved by science in less than 20 years, because we could be growing these things in science, and we'll see them fill the dishes, and then if they crash down, like if you do time lapse, and they grow again, they've responded to stress, we pipette that, and we find that they've undergone molecular evolution. There could be competitions all over the world to do this protobiology, but it'll be like seeing the Earth from space from Apollo 8. You know, that beautiful image? Seeing this is probably how we came into being. And it's a struggle. It's like go team go, you know, because these things are fragile, they fail all the time, but they're collaborating and they're reaching up. So when we hit them with hard ultraviolet, they respond and they turn black. We go, wow, this is our ancestry. We'll rethink our civilization at that point. Certainly our political economy thing, we should. So anyway, that's where this rolls into philosophy.
Very last quick question.
Well, I was going to ask the question, what was the first truly self-replicating thing? But I think what you're saying is there was no such thing, that there wasn't an individuated self-replicating thing. There couldn't have been. That's a stupid question to ask.
And that's where it goes to creationism again, because they talk about this as being the irreducible thing. Well, you don't have to have it. So the little bundle of things, the bundle of molecules when I was 14 that winked, like, you work on it. When I saw it in December of 2013, I was doing yoga and breath work, and I went into a reverie, and I was the protocells. I was just 2.5 hours, I was running the thought experiment. I ran upstairs, wrote to Dave Deemer, my colleague at UCS. He did the drawing, and he wrote back, you found it. You found the kinetic trap. You found the mechanism. And the beautiful thing about that is it's solved. The wink was solved. So I could say back to the dancing molecules, it was little machines all working together that lifted an environment. And out of that environment, that niche, that could come more complex machines that one day could divide on their own. But they did it only within the context of the community to support them. So the whole thing, that's the whole basis. You can't do things without a community support around you. And that could be shown at the protocellular level.
And you heard it here first. Thank you so much, Dr. Damer. He needs his rock back. Betsy has it for you. There we go. So, carrying on with the fantastic theme for this afternoon, talking about small machines working together, is my colleague from the UCSD Department of Pediatrics, Dr. Alysson Muotri, and he's going to be talking to us about cerebral organoids for neurodevelopmental and evolutionary studies. Dr. Muotri.
All right, thank you. Is this working now? Yeah.
Okay, so first of all, thanks Stuart and Eric for the invitation. And so I think this is my first time on this meeting. I'm very impressed with the spectrum of talks, and I promise I'll try to use a language that everybody can understand and not be too focused on the molecular cellular neuroscience. My first really contact with the issue of consciousness was when I was a postdoc at the Salk Institute. I had like some meetings with Francis Crick, and he was the one who implemented the idea that perhaps you can design experiments to understand better how this phenomenon happens. So that was staying in the back of my mind through my postdoc, and then when I assembled my lab, become a assistant professor here at UCSD, I start to use stem cells to model brain diseases. And this is taking in different ways now, and that's the story that I want to talk to you. So when I talk to my colleagues from the cognitive neuroscience department, they are always trying to find when does cognition begins, and you can replace that by consciousness. And there are many ways of studying that, many models. You probably hear like from different models now. But you can grow neurons or brain cells in a dish and just probe, try to understand how they work. Maybe they will assemble a network, or you can move into animal models and ask those same questions, or even study the human brain and see if you can gain insights. I mean, all these models are very useful, and we learn a lot from them. But as any other model, they are simplistic views, simplistic representations of the reality. They all have drawbacks. So one thing that we don't have in neuroscience was a model of the human brain, so you could do like experiments. You can stick an electrode in there and ask and test hypotheses. You can do that in certain circumstances under like surgeries, but it's not really to probe throughout the development in a live human system, live human brain system. So how to get to that model? So that's where we should perhaps start from scratch. And how the human brain appears is throughout the development of human embryonic stem cells. So for those who are not familiar with this system, stem cells or embryonic stem cells can be isolated from the human blastocyst post-fertilization, and you can plate these cells in a condition that they can grow indefinitely with some growth factors in there, and you can withdraw the growth factors or just stimulate the cells with a cocktail of another factors, and they would specialize in the different tissues or different brain types that we have in the body. So that's why we call them pluripotent stem cells. They have this ability to specialize or to differentiate in the different cell types. So there's many ways of doing that. You can get them from surplus of in vitro fertilization clinics. You can transplant somatic cells back into a nucleate egg and clone a human, basically, in a dish. Or more recently, we have the advancement of induced pluripotent stem cells. This was the work from Shinya Yamanaka, who shows that by just overexpressing 4 genes in any somatic cell of your tissue, you can push these cells back in time and have something that is similar to embryonic stem cells. So then we can complete the cycle and actually have in your lab neurons from yourself, for example. So what I'm going to be talking now is one way to create cerebral brain organoids with a novel protocol that we're just developing in the lab that's way more sophisticated and more complex than we ever thought we could get. So I'll mention a little bit of how we do that, how we generate that, and then the activity that we can actually capture from these brain organoids. And finally, I'll show you that we can actually have some networks, some brain oscillations, something similar to what we can get from EEG. And at the end of the day, I'll mention some of the potential applications that we can get from these two in the lab. So the idea is that to create the cerebral organoids, some people will call them mini brains, just because they resemble something that's similar to a fetal brain. But the truth is that this is not like an accurate representation. There are ways to induce the cells to become the entire brain, but we are not quite there yet. Most people are trying to just generate pieces of the human brain, and we have been focusing on the cortex. To drive the cells to the different brain regions, you just need, first of all, to understand the embryology behind it, and then use specific factors that will guide them to the tissues that you want. They grow in the lab inside incubators. So this is more or less a stack of dishes. In each well of those plates, we have hundreds, if not thousands, of those mini brain cerebral organoids. So this is a postdoc working in the lab, and she's one that helped us to develop, to optimize this protocol. She's going to show how these structures look like. These are organoids that probably have like 4 months of age. You can actually age them for more than a year now. And they will be similar to a sphere, a structure with some folding inside that you cannot actually see in this kind of microscope. So people asking me, I mean, can we see that? How big do they grow? So as I told you, I mean, you can actually see them, and they can grow up to like 0.5 centimeter, or a little bit more than that. But after that, because there is no vascularization involved yet, the center of those organoids will actually start to die by necrosis, and the whole system will just fall apart. They will eventually die. But for like 9 months to a year, it's fine to have them. They grow very nicely. And this is visually how they started. The protocol we developed actually starts from single cells. You get these pluripotent stem cells, and you dissociate that in single cells, and they will self-aggregate and form what we call a neurosphere, something that is already in 3 dimensions, but doesn't have the complexity that we want. Most of those are proliferating cells, and you keep them proliferating, they'll just start to reproduce, and some of the cells will become will differentiate in the postmitotic neurons. And then at the end of the day, you just have like a phase of maturation where you just keep them, and they will increase in complexity and size. So if you just replate those cells in a dish in a 2D environment, you can see how they migrate. So here is the bottom of one of these structures, and you see that some cells already migrate out, and they have all these process that looks like radioglia cells, where other progenitor cells will just take advantage of the cells that are already there and will migrate further out, where they will reach to a point where they start to differentiate, and then they would start to form connections. This process is so efficient and is so nice that at the end of the day, you have something that really resembles the fetal brain. This is a cross-section of an actual fetal brain from a human, from an abortus tissue. And this is the organoid that we have in a dish. So it's quite similar. We have a very nice proliferative zone and something that resembles a cortical plate, a cortical dishes in there. We can do a more sophisticated analysis, and all of that is too much information. I'll try to summarize. So all these colors are immunostainings with different specific antibodies for the brain regions of the cell types that we want to investigate. At 1 month of age, most of the cells, they are proliferating cells. They are nesting positive Ki67 cells. So these are just progenitor cells that are replicating. By 2 months, we start to see some of the deeper layer cortical neurons that we start to appear. Markers such as CTP2, this is the layer 5 and 6. We start to have some of that. There is still some proliferative zone in the center of this organoid, and that's the time where we start to see some of the folding on these organoids. By 4 months, the whole structure will change a little bit. We start to see a higher proliferation of glia cells. The folding will become more common, and the organization of the cortex become more and more sophisticated. So this is all good news. You can follow that by cross-section of those organoids, and you can actually record live cells, how they migrate and differentiate. Or you can do, like, again, immunostaining just to see the different markers, something that look like the different cortical layers that we see in the human developing brain. One thing that I mentioned to you, that between 4 and 6 months, the glia cells, mostly astrocytes, will start to take over the proliferation, and they will become about 50%, a little bit higher than that, of the entire cell population. At the same time, I mean, we see an increase of the different markers for the different cortical neurons, subtype of neurons. Another way to analyze the nature of those cells and the diversity of cells that we have is to capture individual cells into a microfluidigm system like that one. So individual cells will be captured, and then we can probe the gene expression of that specific cells. And we can do that by target gene expression, designing primers of way to check specific genes, or using global unbiased gene expression, just look at what the cells are expressing. That can tell you a little bit about the identity of those cells. So this is just some of the population analysis. This is a violin plot, so the shapes represents the amount of the population, and here is the intensity of the gene, or how much that gene is being expressed between 2 and 4 year old. So GFAP is a marker for astrocytes. In the beginning, we don't have anything in there, a little bit of cells, but as they mature, you see that the population of glia cells really becomes more apparent. And that is what we think will guide the amount of electrical activity that's coming from these organoids. So this is another way to represent the cells, and we can do, again, unbiased gene expression, and then we have these maps, and this is the map of the entire cell population that we have in one organoid. Depending on the identity, you can start to label them. So these would be the progenitor cells. There is some GABAergic cells, glia cells in there, and most of them are glutamatergic cortical neurons from the different subtype of cortical layers. So what about activity? One way to simply visualize if there is activity going on is to using voltage sensitive dye. So you can actually record when these neurons are firing using some of these dyes. So I hope you can appreciate that there are some neurons that are just being active now. This is just by looking at the entire organoid with a high resolution fast camera. So you can visualize the spikes in there. So another way of doing that is just placing the entire organoid, the entire ball on top of these multi-electrode arrays. So those images that we have here, these are 8 by 8, 64 electrode organoids on the bottom of a dish, and then you plate those organoids on top, and you can see that they stay consistent in the same place over time, and you just record their activity. So at the end of the day, what we have, it is a raster plot for each channel, and we'll give you, each trace would be like a spike. If you have too much of those, we call them bursts, and when the bursts fire together, we call that a synchronization index. So we have been looking into the evolution of those brain organoids over time, and we compare to other species. For example, this is if we do the same thing with mouse. Most of the activity of the mouse activity that we can get with this system, it's here in the bottom. This one is just a representation of a brain slice. So if you just take a piece, a chunk of the mouse brain and put on top of these electrodes, you're going to have this much of firing activity. So in green here you have other protocols using human iPSCs to differentiate these cells into neurons. Most of those protocols are in 2D, but we do have like one 3D protocol here that increases a little bit the level of activity. So what we have now is just the representation of the different time points that we have been collecting to show the evolution of these cortical organoids. And to our surprise, we have something that it's quite far from the 2D system. And again, with turning points, kind of accelerate evolution of the number of spikes between 4 and 6 months of age. So what happens during this time? So this is time point, 2, 4, 6, 8, 10 months. And this is some parameters that we can trace. For example, number of spikes. As I said, the number of spikes will increase over time. The same is true for the burst frequency, when lots of spikes fire at the same time. As a consequence, the distance or the velocity between the electrodes will decrease over time, and will increase the network frequency, showing how much of those are firing at the same time. So it increases. Again, it's all indications that the complexity it's in there. It's quite striking when we compare the 2D system, and this is the 2D system over time. This is the maximum we can keep those neurons alive in a 2D system. After that, they would just detach and die. But as I said, with the 3D brain organoids, we can keep them alive for a longer time. And the synchronization index, it just goes up and then finally reaches a plateau, which is the amount of electrodes that we have in a dish. So it's all connected at the end. So just to prove that everything is correct, we did some pharmacology. This is when we use specific drugs to try to understand how our network is assembled. For example, we can use inhibitors of electrical synapses. And when we do that, we reduce completely the number of bursts or the electrical activity that's in there. Or you can use inhibitors of GABAergic inhibitory neurons just to see how the networks behave. So as a summary of all these pharmacology experiments, it again is another way to show that we do have inhibitory neurons that's coming from the same population of excitatory neurons in our dish. But most of the cells are really excitatory glutamatergic neurons. So as we grow those neurons past 6 months up to a year, we see something that was quite new, that was never reported, is this amount of complexity or information that we can gain over time, to the point that we can see that these network oscillations that just appear, this is something that most of my colleagues didn't even believe that was possible to do in a dish, thinking that we might need something else. But the truth is that, no, we can do it, and we do it in different ways. I mean, the first of all is we use the same information that we can get from the electrical activity, and we do the spike extraction here, but we use a low pass filters to get for what we call the local field potential. And we correlate the synchronization spikes that we have with these LFP to confirm that these oscillations are real. And as expected, we see that the brain oscillations actually appear to the time point where these connections become more apparent, more clear, which is about the 4-6 months of age that I just told you. And the complexity happens over time, so these oscillations become again more apparent, passing these 4-6 months of age. The same is true for the voltage oscillations as well. And we did a bunch of analysis just to show that how this complexity actually appears. And one thing that we notice is that it reaches a time where there is everything there, but then, I mean, it decreases a little bit. It's almost like you cannot cope with that much information. Or our system, being an artifact in culture, we can really, there's probably cell death going on, something that we haven't investigated as well. But the take-home message is that there is a peak of activity and complexity, and then slowly it seems to fade out. Another explanation for this idea that the level of complexity does not increase is the fact that by in old cerebral organoid, we start to see these irregular oscillations. So before these 4-6 months of age, everything was kind of very similar. Each organoid was behaving in a very similar way to each other. But after passing this threshold, they kind of go in different directions. They are more diverse, and maybe it's the medium or the average of that diversification that suggests us that this complexity is no longer there. We don't know what happens to each one of them, why each one of them will actually behave in a different way, but this is something that we observe in a dish. And finally, something that we are trying to do is, again, taking advantage of these oscillations that emerge spontaneously in a dish and correlate with EEG from people. So we are using induced pluripotent stem cells from individuals with epilepsy, for example, and then trying to correlate that with their EEG pattern. So if we are able to correlate that in a very nicely way, it means that we can now translate everything that we learn from these brain organoids to that person. That would be helpful, for example, to predict responsive to drugs, antiepileptic drugs. That would be one application. So some people ask me if this is all about the 3D culture. So one experiment that I think is quite important, it is if you get one of these brain organoids, dissociate, and try to reassemble them back, we don't see the emergence of this brain oscillation. So it's something that requires developmental time to reach out. You cannot just assemble the pieces once they are formed, so they will never get to that point. So we never see brain oscillations with artificial neurospheres that we create from dissociated single neurons. So as I promised, this is just some of the applications that we do have going on in the lab, and just would like to explore that a little bit. My lab has been focusing a lot on autism and try to understand the autistic brain. So the model we have now is really to reprogram cells from a group of autistic people, a group of controls, create these brain organoids in a dish, and look for differences. Those differences can be at the cellular level, at the molecular level, or at the network level. This is what we are more interested in now. We think that most of those differences will actually happen at these oscillatory network levels. So this is exactly what we are trying to achieve now, and perhaps using that system to do what we call personalized medicine. There is a group of genetic mutations, variants of genetic mutations that might be responsible for those cases. So if you can find better treatments, not only for the core of autism, but perhaps comorbidities, that would be something very useful for these families. So this would be like the more practical application of the system. So another thing that we are using these organoids is to probe and understand infectious agents, for example, the Zika virus. This is something that we actually use to prove that the Zika virus causes microcephaly. We expose these mini organoids to the Zika virus, and we see that it goes and infects exactly these neuroprogenitor cells. As a consequence, the cortical thickness is reduced, and you see lots of deficiency in the number of different subtype of cortical populations. So this was interesting to see. I mean, it can easily explain the macrocephalic phenotype of those babies. But another thing that we did, and that's quite scary to me, it is if you use a low MOI, a low amount of virus to infect these brain organoids, we don't see the macrocephalic phenotype. This is because the cell death is more subtle, and the virus is affecting the neurons in a different way. As a consequence, what we have is an aberrant network formation. So one can expect that even the kids who are infected by the Zika virus, but do not show the macrocephalic environment, they might actually behave in a different way. They might actually become autistic or have seizures later in life. So we can use the system now to predict something that infectious agents may actually do to the brain. And this is not only true for infectious agents. We are actually using the model as a probe to see different environments, different toxins that you have in your environment. So every day we are exposed to more and more new chemicals in our environment. This is true for pregnant females as well. So what happens when those toxins actually reach out to early stages of brain development? So now we have a system, although artificial, with no like membrane to protect the brain, something in direct contact, but nonetheless, it might put a red flag in a specific toxin that it's out there. So the idea is to create something that's called the brain-friendly label. So before something goes into the market for a pregnant female, we're going to be testing this model and make sure that does not affect brain development. So another application for another environment, and this is something that I'm doing with Eric here, is to send these brain organoids into space. So we can see the effect of microgravity or space radiation during brain development. So we hope that we can send the first brain organoids this year to space, and we'll see how it goes. Another thing that we can do is, this is something that I'm also very excited, is to compare these brain oscillatory oscillations from the organoids from non-human primates, such as bonobos and chimpanzees, and see how much they differ from us. One thing that I can tell you right now is that the sophistication of those networks, it reaches to that level at much faster pace compared to humans. At least for the time points that we have, any other species will get to the level that we think it's already like complex 2 or 3 months before any humans that we ever tested. So there is a reason for this a long delay of the human networks to reach out to that level. And we don't know what it is, but it seems like very human specific, very modern human specific. We are also taking advantage of those cells and using genome editing to modify some of the candidate genes for human selections that make us different from other hominids, for example, the Neanderthals. So we can Neanderthalize one of these mini brain organoids and study their minds or their networks in a dish. So see, this is another way to study evolution using that system. So I think I spoke too fast, but I'm glad that we got through that. So I'll thank members of my lab and my collaborator here. This is Brad Wojtek at UCSD, who is responsible for all this brain oscillation analysis, and obviously the families and subjects that we use in our research. I'll be happy to take any questions. Thank you.
Thank you so much, Dr. Muotri. So I got to ask you, have you made any from your own stem cells?
No, I avoid doing that. I don't want to see it.
All right. Questions down there at the back there?
So this is super interesting. Have you tried doing these cells from the beginning in a 3D gel?
We try. And they don't develop with the same consistency as if we just leave them floating around. If we put in a Muotri gel or something, there is a physical barrier.
So when I say 3D gel, I'm talking about doing your 2D, putting the cells, and then putting another layer on top.
Oh, I see what you mean. Engineer the layers.
I think you'll get even better results.
Yeah, we did that. Yeah.
Oh, you did that. What happened?
We did that. At least for the system that we use, we could not make the layers to connect. So we may have to modify the type of matrix that we use. But that's right on. That's exactly what we should be working on.
Talk to me in abyssal, because I think you might get even more complexity. It's really worth trying.
Okay.
The one behind there.
Hi there, great talk. Quick question about the missing heritability in autism. So you sort of alluded to this with your CRISPR model at the end, but Evan Eichler and others at the University of Washington have alluded to the fact that many of these phenotypes are a result of structural variation, like the 16p variant, and a lot of these inversions, small deletions, but mostly duplications in a lot of these. So are you moving in that sort of direction? And do you have, does that affect arborization and so on and so forth?
Yeah. We actually published on some of those variants before, and we do see alterations in neuronal arborization, for example. This is one of the first phenotypes that we look for, just brain morphology, neuronal morphology. But that results in a defective network as well. What I think is promising is that most of the phenotypes that we have could be reverted with some drugs. By just exposing those neurons to certain drugs, you can revert that back to the neurotypical state. So I think that's encouraging.
And just a follow-up. And then for gene duplication regions, like you were alluding to, thinking about evolutionary trajectories, do you have any, you or anyone else know of methods that are moving in that direction?
Yes, we have, for example, one disease that we modeled before, and this is all published, is a condition called the MECP2 duplication, where the MECP2 gene, which is one of the candidates for Rett syndrome, instead of getting deleted, it gets duplicated, and it causes a severe neurological disorder. So we characterize those neurons, these brain cells, in addition. They do have some defects as well. That can be reverted with some epigenetic drugs, but most importantly, we designed a CRISPR enzyme to go and knock down one of the copies of the MECP2. So now we have the corrected balance of the gene, and that was enough to revert the condition as well.
Okay, very cool, thanks.
Right up here.
So have you had the opportunity where you have two of these organoid colonies, one of which is synchronous in its oscillations or beating or whatever it is, to show any synchrony between nearby but physically separated colonies?
Yeah, that's a great question. We haven't done those experiments, but it's definitely on my list to do. Yeah, perfect.
Yeah, so when we're looking about mathematical creativity, one of the things that we are expecting to happen in neurons is they protect themselves from going into oscillation, because otherwise they need to avoid going into too much oscillation in order to do certain mathematical things. Now, it looks like that's what you might find that happens. They either die or they develop a protective mechanism, and then their oscillation starts to go down. So is it possible to see more of the exact pattern of what they're doing, in detail, to see whether they're doing, what they're doing, which way they're going?
So I think to do what you are proposing, we should be studying individual ones. And we do have this data. I'll be happy to collaborate with you. Yeah.
Beautiful work. I was wondering about your thoughts on other cells that are present in brain. In brain, neurons to astrocyte ratio is 1:7.
Yeah.
So what do you think, if you include those astrocytes in your model, what you would predict? And in case of autism, your thoughts on vaccination and autism?
Oh, okay. Two questions. So the astrocytes, they are actually present. They start to kick in about 4 to 6 months. And that's exactly the time point that we see the starting of the appearance of those brain oscillations. So one idea that we have is that the astrocytes are the one responsible for the emergency of those oscillations. So the experiment we are trying to do now is to knock down the astrocytes so they will never appear over time, and we'll see how the oscillations look like. So we think they are causal. They are leading to the oscillations. Without the astrocytes, only neurons will never do it. You need the glia cells. So autism and vaccines, as far as I can tell, I don't study that specifically, but this link it has been debunked by science like 2 or 3 years ago. So I don't think there is a causal link between autism and vaccines. Yeah.
Okay, last quick question.
Could your oscillation phenomena actually chaotic attractors?
That's a great question, which I think is linked to that experiment that we should do it. Yeah, I don't know.
Yeah, well, I suspect it is.
Okay.
And it will be interesting interpretations.
Okay, great thought.
Very good. Well, saving the best for last. Thank you so much,
Thank you.
Dr. Muotri. So such a fantastic session. So I would like to, personally to thank the conference organizers, and I see Abby in the back there. I think we owe Abby a huge thank you. Woo! And without further ado, our friend Stuart Hameroff. Stuart, your friends.
Thank you. Thank you, Eric. Thank you all for being here. So I'm going a little bit out of my comfort zone, which is normally in the brain, consciousness, microtubules, although I will talk about microtubules, and especially after two experts in the area. And as my friend David Chalmers said, people generally don't like your quantum consciousness ideas. They're going to hate your quantum evolution ideas. So bear with me. And I don't think what I'm going to say conflicts with Darwin, nor with what Bruce or Alysson said, Bruce in particular, since we talk about origin of life. But we'll see. So the quantum pleasure principle, did life evolve to feel good? Let's first ask the question, when did consciousness arise? And here we have a history of the universe from the Big Bang, at least this eon of the universe, up until fairly recently. And being is meant to imply the origin of the onset of consciousness in the universe, and some people think it was fairly recently with the onset of tools and language. Some people think it was with the onset of animal life, with eukaryotes a couple billion years ago. Some people would say with first life, any type of life. And some people would say before the Big Bang, even. Eastern spiritual people, Colin McGinn had this idea a few years ago. And Roger Penrose, who was here earlier in the week, in his book Cycles of Time, of course, talked about serial eons of one universe where our Big Bang was preceded by another Big Bang. So was consciousness in previous eons? Well, obviously, we don't know the answer to that as yet. So when did consciousness arise? Let's first focus on the question of life and consciousness. Which came first, consciousness or life? Now, most neuroscientists, most psychologists, most Western philosophers would say that life preceded consciousness, that consciousness emerged from life. You've heard the story, brain computation, complex computation, then at some point consciousness emerged. That's pretty much the dogma, the standard party line in neuroscience and philosophy. On the other hand, we have panpsychists, and I must say that some of the leading lights in consciousness theory, like Dave Chalmers, Christoph Koch, Giulio Tononi, now resort to panpsychism because they can't really crack the hard problem. They can't explain how qualia, how subjective experience, phenomenal awareness, and that sort of thing comes out of computation, because they're quite different. A point Roger made quite nicely earlier in the week, that understanding, for example, is not really a computation. You have to be outside the system. Naturalism, Eastern philosophers, spirituality, and we heard Deepak talking about this earlier. Whitehead, Alfred North Whitehead, and Rogers' objective reduction would say that consciousness is intrinsic to the universe, that it's somehow been here all along, if you will. And if that's the case, if consciousness was here all along at the time of the onset of life, perhaps life and somehow emerged from consciousness or related to consciousness or used consciousness in some sense. Let's next ask the question, what is life? And there are several approaches. I'm going to give you 4 or 5 different approaches. The first is functionalism. For example, Lynn Margulis Sagan, who is a great biologist, talked about self-organization, metabolism, growth, adaptation, response to stimuli, replication, reproduction, and evolution. But these are functions. They're not really intrinsic causes or the nature of life, and they occur in other systems that are not living. For example, computer programs, video games, and weather patterns. For example, this is a picture of the Great Red Spot on Jupiter, which is more or less stable but evolving over time, and it's been there decades at least. There's also the possibility of vitalism. In the 19th century, there were many scientists and philosophers who talked about a unifying energy field, a life force, or élan vital, pervading living systems. And these were based either on electromagnetism or forces outside science, mysterious forces, unknown forces. But when molecular biology came along and reductionism seemed to succeed, and DNA and so forth, there was no need for vitalism, it seemed, and vitalism became taboo and remains so to some extent. Also, there are a group of descriptions relating to mathematics, scale invariant logic, information flow, complexity, coherently pumped metabolism, emergence theory, self-organized criticality. We heard about that earlier from Stuart Kaufman, Paul Nurse, Paul Davies, Jack Tusinski, and Travis Craddock, the latter two collaborators of mine who have talked about this. But non-living systems have these properties also. Also, there's what I would call quantum vitalism. In his book in 1935, the famous quantum physicist Erwin Schrödinger suggested that life's unitary oneness derived from quantum coherence, and that life and that memory, for example, was stored in aperiodic crystal lattices. But many people said subsequently that biology is too warm, wet, and noisy for functional effects in delicate quantum systems. If you want to build a quantum computer in the laboratory, you need to do it at absolute zero to avoid jiggling and decoherence. Or is it? And now we know, for example, that plants use quantum coherence in photosynthesis to make energy and food, and utilizing superpositioned electron excitations, excitons, propagating through chromophores, 7 or 8 chromophores in this FMO protein to get from the light harvesting complex to the reaction center. And the electronic excitations appear to go through all of these chromophores simultaneously in superposition. And this results in an efficiency, a tremendous efficiency, so that the light energy is converted to food with a high efficiency. And without this high efficiency, we may not be here, because this makes food that animals eat, and we eat the animals or we eat the plants, and this has allowed life on Earth to flourish. And so we probably owe our existence to quantum coherence in plants. And as Honorbond showed this morning and in previous publications, microtubules also utilize quantum coherence in ways that I'll mention a little bit later. Great minds think alike. A number of scientists that I studied when I was younger and still rely on had similar ideas that life involves some kind of electron resonance, pi electron resonance, and quantum effects. The Nobel laureate, he got it for something else, Albert Szent-Györgyi, said that the essence of life is in submolecular electron mobility, in other words, within molecules. The Russian scientists Philippe and Albert Pullman attributed life to quantum behavior of electrons in pi resonance clouds, and pi resonance clouds are the basis for organic chemistry. We'll come back to that point. And Herbert Froelich, in a series of papers in the late 60s and 70s, said that life in coherent electron dipoles in nonpolar regions in geometrically constrained proteins were essential to life, that these would oscillate coherently, being pumped by heat, basically, because of the geometrical constraint, and the nonpolar pi electron resonance clouds would pump coherently. He had attributed it to membrane proteins, and I had the good fortune of meeting Froelich in the 1980s, and I suggested that it may occur in microtubules, and he liked that idea. In fact, he invited me to contribute a chapter to his book, which I did. Another possibility that I'm going to raise here is that life may involve intrinsically feelings and what we might call proton consciousness, which eventually leads to consciousness. And I'll explain why shortly. And thus far, at least, only living systems have consciousness and feelings. And so they may be intrinsically related. But that raises the question, what are conscious feelings and what is consciousness, which is why we're here in the first place and why we've been having these conferences all these years. And we're trying to move closer to an answer. So let's go back to the previous question I raised. Which came first, consciousness or life? And I want to focus on the second area, panpsychism, naturalism, Eastern philosophy, spirituality, Whitehead, and objective reduction. And specifically, skipping over a lot of other stuff, get to Roger Penrose's idea of objective reduction. And Roger was here early in the week, and most of you, I hope, had an opportunity to hear him speak, both here at the conference in a workshop and also at UCSD in a special talk. So Roger addressed the measurement problem in quantum mechanics in a great book that I recommend, The Emperor's New Mind, that influenced myself, James Tagg, and many, many others. He took issue with the idea that consciousness was a computation and that AI would reproduce consciousness by sheer brute force computation. And he argued through Gödel's theorem. And what was missing, he argued, outside of computation, was something that involved quantum physics and specifically a solution to the measurement problem in quantum mechanics of why we don't see multiple coexisting possibilities, the problem of Schrodinger's cat, for example. And to do that, he made a leap, an intellectual leap that is still astounding in relating quantum physics to general relativity. And this could be a whole lecture in and of itself, of course. And to do so, he used two-dimensional space-time sheets. And Jonathan Schooler, in his excellent talk this morning, also showed two-dimensional sheets. But Roger brought in general relativity. So in a two-dimensional sheet, you have 3 dimensions of space are condensed to 1 dimension, and you have 1 dimension of time. Now, Einstein showed that matter was essentially equivalent to curvature in space-time geometry. And this was applied to large systems. And famously, the idea that a star behind the sun, the starlight would be bent by the space-time curvature due to the sun and could be seen on Earth, even if the star was directly behind the sun. And Sir Arthur Eddington, in a famous experiment around 1920, I think, 29 maybe, at the top of a mountain during a total eclipse, was able to see a star that was known to be behind the sun, proving that the mass of the sun causing curvature in space-time bent the starlight around so we could see it on Earth. Well, Roger applied this idea to small things. That was a big thing, big mass, big curvature. He said that particles, any particle, would be equivalent to curvature in space-time, and a small particle would be a small curvature. So, for example, here on our two-dimensional space-time sheet, here's a particle here, and here it is, the same particle in a different location, and the curvature has changed. So if you think of this particle oscillating back and forth between 2 locations, the space-time curvature is oscillating back and forth. So this would be one particle oscillating back and forth between 2 locations. Now, to address the problem of quantum superposition, because this still remains a fundamental question, how can something be in 2 places at once, or 2 states at the same time? Quantum superposition. And a lot of quantum physicists deal with this without coming to grips with the basic question of how is this feasible in the first place. And Roger said it simply means, well, not really simply, but it means that there are both curvatures exist. So this curvature over here and this curvature over here. So you have kind of separated curvatures, and the particle, the same particle in this case, is in 2 places at the same time. And so here we see the two particles and the 2 space-time curvatures in a quantum superposition, and you might this could collapse to one or the other. Now, you could imagine that if each curvature continued, the space-time which it represented, or which represented it, would evolve its own universe. And this is known as the multiple worlds hypothesis. And this is actually the most popular view in quantum mechanics among quantum physicists, because it solves a lot of problems, even though it leads to the rather extensive metaphysical baggage of an infinite number of overlapping universes. But this would be the natural extension of that idea that each space-time curvature would develop its own universe, and you have multiple worlds. But Roger suggested that rather than that happening, the space-time, the superpositions or separations in space-time are unstable. and will self-collapse, that is, undergo objective reduction. That is, they will undergo reduction due to an objective threshold, therefore objective reduction, or OR, at a time t given by this very simple equation, t equals h bar actually over E sub g. It's basically the indeterminacy principle. So at time t, a superposition separation of E sub g, and you can calculate this from the mass separated from itself or the space-time curvature separated from itself, and predicted at time t that self-collapse or objective reduction would occur. So here's time t. This should be at this dotted line. T equals h bar over E sub g. H is Planck's constant, and h bar is Planck's constant over pi, or the Dirac constant. And at this time t, this particular superposition, this possibility will cease to exist, and this one will continue. So the wave function has collapsed, you might say, to this possibility, and this one no longer exists. And the kicker was, he said, that this would be accompanied by a moment of conscious awareness, bing, at this particular moment t, at this time t. And that this bing, you might say, would have some component of feelings, of awareness, shown here by this emoji. And this has been a great advance in consciousness studies, bringing emoticons into to represent qualia. Thank you. And these events would be occurring in space-time geometry everywhere around us, all the time. But they would be random and disconnected, and what Roger calls proto-conscious. So this is essentially equivalent to decoherence, you might say. Decoherence is the idea that superpositions are occurring but go away due to interaction with the environment, but there are problems with that. But nonetheless, and it may seem strange to think that collapses everywhere are causing these little moments, but it's no stranger than panpsychism, for example, which says that consciousness or some kind of awareness is everywhere, attached to the lectern, attached to everything. But they would be proto-conscious, disconnected, random, not entangled with others, not have no memory. So they would just happen, and we have no consequence, you might say. But they would be occurring in the universe all along. Now, the choices in each OR event are not necessarily random, in which case they would follow the Born rule in quantum mechanics, but would be influenced by what Roger called Platonic values embedded in space-time geometry. And this would apply only when the superpositions, when E sub g got quite large and the system was organized or orchestrated. And it wouldn't necessarily apply to proto-conscious values occurring randomly here, there, and everywhere, but when the system evolved and became more organized or orchestrated, we might get non-computable effects leading to creativity, the kind of thing that James Tagg has been studying and talking about, and implying creativity and even inspiration, you might say, from information embedded in the universe. And this mechanism, although it's not, most neuroscientists, philosophers don't really take it seriously for some reason, nonetheless, it is the only specific mechanism for consciousness ever proposed. Everything else is kind of hand-waving, emergence, complexity, it just happens. Or get rid of it altogether, like Dan Dennett. It's the only mechanism that, and I've been following this field for a while, that's ever been, specific mechanism that's ever been proposed. So let's go back to our history of the universe. And note that at the beginning of life, that consciousness, that these proto-conscious events may have been happening all along at some point, maybe subsequent to the Big Bang, and have been present in the universe when life began. So Bruce's excellent talk gave some another idea, which I think is consistent with this, and maybe we can talk about it later. But I'm going to talk about the primordial soup, and I think it could be also in the kind of heated pools that he was talking about. But the idea of the primordial soup was proposed in the 1920s by Oparin and Haldane, and it was described as a simmering mix from which biomolecules emerged 3 to 4 billion years ago. And here we see a picture of what it might look like with some globules indicating oily materials as well as water. So in the 1950s, Stanley Miller and Harold Urey at University of Oregon simulated a primordial soup and found organic amphipathic molecules. So they had a system simulating Earth's primitive ocean. They had an atmosphere, cloud formation, sparks simulating lightning and energy, and with some power. And they ran this experiment, and what they found was organic amphipathic molecules. So organic amphipathic molecules, the precursors of life. So amphipathic molecules are those which have a nonpolar end, such as these aromatic rings, a six-sided benzene or phenyl ring with three extra electrons that's nonpolar or oil-like. And you know oil and water don't mix. And a polar tail that would have a charge like an OH and an H over here, and nonpolar and polar. And these are very similar to dopamine. Dopamine is very much like this. Dopamine, the reward molecule, the pleasure molecule, or serotonin, the mood molecule. And as I'll show you in a minute, all psychoactive drugs pretty much have these pi resonance clouds, or nonpolar benzene-like rings. So these nonpolar oil-like rings, which are made of pi electron resonance clouds on one end and polar water-soluble tails on the other. So a little bit about the discovery of these type of structures, exemplified by benzene. It was known in the I think the 1800s by chemists that they knew about the alkanes, hydrocarbon chains that could be as arbitrarily long with a formula of CnH2n plus 2 with all single bonds. Alkanes, which had one double bond with CnH2n. And here's longer alkanes, and here are longer alkenes with a double bond. And that all made sense. But then they had a compound. They knew the structure was C6H6, and they knew it was different. It was oily and flammable, but it had C6H6, so it didn't quite fit. And Kekulé, who was Dutch or German, I forget, a chemist, had a dream one night of a snake swallowing its tail, the Ouroboros. And he woke in the morning and said, benzene, C6H, must be a ring structure in which the snake has swallowed its tail. And there are three extra electrons. You can see here the electron double bonds, and they be shown here for simplicity, get rid of the hydrogens, and we see this benzene ring with three extra electrons. And these hexagonal benzene rings have the extra electrons are delocalized, and they're in the pi orbit, so they're called pi orbital electrons, or pi resonance among the six carbons forming pi resonance clouds, which are conducive to quantum effects. Now, if you put in a bulk, it's like it's gasoline. You put it in your car and your car runs. But if you have these spatially separated, you get interesting quantum effects. So the extra electrons form these electron clouds above and below the ring, and so you basically have a quantum object here. And this supports electric and magnetic dipole oscillations, excitons, charge transfers, phonons, and fluorescence. And we know that anesthesia, for example, acts in regions like this, and I'll come back to that point. So this raised the question: since do life and consciousness derive from pi resonance in biomolecules? And the evidence from anesthesia acting according to the Meyer-Overton correlation that Travis Craddock spoke about earlier would suggest that it does. So these pi resonance clouds attract by van der Waals attraction to a van der Waals radius, and once they get at just the right distance, and they do this themselves because that's where they're most stable, the three electrons form an electron cloud, and this electron cloud sort of repels this electron cloud, and oscillates back and forth. So you tend to get an oscillation of these electron clouds in terahertz. So these oscillations here would go back and forth in terahertz, 10 to the 12th cycles per second. And you can also have quantum, oops, sorry, wrong button. Gave away my punchline there. Yeah. So there it is. Pi resonance clouds are rated at a critical distance oscillate in terahertz, 10 to the 12th hertz. And these appear essential to life, consciousness, and anesthesia, as I just said. And we can also have quantum superposition. Now, as I said, and as we know, oil and water don't mix. So if we have these amphipathic molecules, the nonpolar rings are going to attract by these van der Waals attractions and come together and then oscillate, and the polar chains are going to stick out. And this forms what Operin called a micelle, in which you have a nonpolar interior and a polar exterior. And this became the basis for pretty much all biomolecules. For example, proteins fold in this way. Protein folding is driven by nonpolar pi resonance groups of aromatic amino acids, tryptophan, phenylalanine, and tyrosine coalescing in the middle, and the polar groups sticking out into the water. So because the pi resonance wants to get away from the water, so the polar groups interact with the water and stabilize it, so the nonpolar groups can be shielded from it. And this is how proteins fold. This is with a cartoon, obviously, of three of such these rings. These can be more complicated. For example, these are micelles that have been calculated and modeled by Arvydas Tamulis in Lithuania, with many of these pi resonances inside a larger micelle, isolated from the polar environment, and allow quantum stuff to happen inside, despite being in a general polar environment. And the same thing happens in membranes. For example, in lipid membranes, cholesterol, for example, the nonpolar regions go in the center, the nonpolar and the polar stick out. So cell membranes have this basic structure, with the polar group sticking out of the cell or into the cell, and the interior of the membrane being nonpolar and shielded from the polar environment. And also in pi stacks in nucleic acids, DNA, and RNA. I think Bruce was talking about RNA emerging early in life, and it also has a pi stack right down the middle of DNA and RNA. So proteins, lipids, and nucleic acids, including DNA and RNA, all have nonpolar interiors of pi resonance groups, something that Travis, Jack, and I have described as the quantum underground in living systems, because it's at the core of all biomolecules in all of our cells. I should also mention that all psychoactive drugs, it seems, have these pi resonance groups, including dopamine, as I showed, serotonin, which has the indole ring of a five-sided and a six-sided, and the psychedelics have more elaborate pi resonance structures, shown here in LSD, DMT, and psilocybin. So these pi resonance groups are everywhere that you might expect to see subjective experience. So back in the primordial soup, let's go back to a few billion years ago and imagine what might have happened when, what might have happened when these pi resonators came together in the middle of a micelle, if you will, and eventually they would, if they were isolated, they would reach threshold for Rogers objective reduction at time t equals h bar over e sub g, and here would be the space-time correlate of that. And this is shown for 3, but this would involve many, many, many more to have this happen in a reasonable time. So this is just a cartoon. But at that moment, there would be a moment of, at t equals h bar over e sub g, there would be a moment of proto-conscious awareness, bing, if you will. And there it is in the protein, or in the micelle, and also in space-time geometry. So these qualia, if you will, or feelings of primitive, the feelings associated with these primitive OR events would be random. but some would be positive. That is, they would feel good. And there's our smiley face. Now, some would also feel bad. But this could provide a feedback fitness function and system that you can imagine the micelle getting larger and developing more. And since there was pleasure involved, and also displeasure would be go the other way. They would want to avoid displeasure. That this could somehow drive the development of these micelles to elaborate a greater structure to optimize pleasure. Now, it turns out these pi resonance rings have two stable arrangements. There's a sandwich where they're sitting on top, but that's metastable. The T shape where you have one basically perpendicular to another, and then parallel displaced where they're sandwiched but offset. And these are the two stable states of this system, of this simple system. And we can just imagine that T shape is displeasure and parallel displaced is pleasure. Now, Hartmut Nevin, who was here earlier in the week from Google's quantum computing group, Quantum AI, suggested that relaxation of a system to a stable state might be pleasurable, and relaxation of a system to a less stable state might be displeasure or pain. And relaxation could also apply to quantum collapse, as he said. So one possibility is that a system that collapsed to something like this at the bottom would be pleasurable and would provide feedback for the system to optimize this type of arrangement, and something that collapses something like this would be avoided. So this would be a feedback fitness function for these systems to develop and evolve to optimize pleasure and avoid displeasure. So with pleasure as a feedback fitness function orienting these pi resonance groups, I raised the question, did life then evolve to orchestrate and optimize OR-mediated pleasure? And I'm calling this the quantum pleasure principle, something that Dave criticized severely, but that's okay. And I'm in good company to some extent with Freud, I guess, who wrote Beyond the Pleasure Principle, and this is definitely going beyond the pleasure principle to the quantum pleasure principle. And again, I repeat that Rogers' OR mechanism is the only mechanism for qualia ever proposed, other than just hand waving. So, did my cells then develop and evolve? My cells then developed and evolved to proteins, lipids, and nucleic acids. So let's pick one particular protein, which happens to be my favorite protein, tubulin, the component of microtubules. And it turns out that, and this is work by Travis Craddock and Jack and I and others from 2012 in PLOS One, where he mapped all the pi resonance groups in tubulin. And tubulin is 110,000 molecular weight protein, and it has 86 pi resonance groups composed of tryptophan, phenylalanine, and tyrosine. There are 8 tryptophans, and the rest are phenylalanine and tyrosine. And in the red, you see where anesthetics bind. It turns out that anesthetics bind to tubulin and quite likely exert their effects erasing consciousness on tubulin, as Travis told us in his talk earlier in the week. And so 86 of these, and these are interacting. So if you think of the two pi resonance interactions, the T or the offset P, we have 2 to the 86 possible such interactions. So rather than just one smiley face and one frowny face, each tubulin might have a whole cacophony of feelings and qualia in one tubulin. And then you have about 1 billion tubulins per neuron. So these tubulins would then have, and I've written a chapter about this, and I'd be happy to send it to anybody. The tubulins then coalesce into little sheets and then into cylinders and forming microtubules, which are sensitive to all kinds of energy. All the ambient energy available are microtubules are sensitive and can harvest this energy and drive it. Ultraviolet energy, terahertz, and cosmic microwave, gigahertz radiation would all drive oscillations and resonance and provide energy to microtubules, which, as we see here, would reach a critical threshold at time t for Bing. to occur at time t equals h bar over E sub g. And the microtubule was born. These resonances led to orchestrated OR, further optimizing pleasure. And this would occur due to something happening at the level of space-time geometry much below it. So by orchestration, I mean the following. If you think of proto consciousness as something simple, random, and disconnected, it's kind of like if you go to the symphony and the orchestra is warming up and each musician is practicing his or her instrument, and you hear a cacophony of sounds, different tones. It's really noise. So that would be like proto-conscious events occurring everywhere. They're not really connected. And then the orchestra begins to play and you get music. So the point of the microtubule and biology in general would be to organize and orchestrate these proto-conscious moments into consciousness and to give turning noise into music, you might say. And in fact, I think consciousness is more like music than computation, and the brain is more like an orchestra than a computer. Now, there's some evidence for these oscillations. This Anirban showed this slide earlier today, looking at neurons at the level of neurons, individual microtubules, and tubulin, and seeing, starting at the bottom, at the fast end, terahertz, gigahertz, megahertz, and then going up to microtubules, gigahertz, megahertz, and kilohertz, and then megahertz, kilohertz, and hertz. So, and at each one, as he told us this morning, he sees these triplets of triplets. So we see this kind of multi-scalar hierarchy in microtubules and neurons and all cells, for example, maybe in Alysson's organoids, and I don't say why not actually, from terahertz to up to much slower. So we've developed this model more or less based on this idea. So here we have a neuron, a pyramidal neuron, which has these interrupted, disconnected microtubules. Here we see, as we go down in scale to a microtubule, and then look at the tubulin pathways of adjacent tubulins, and then down to the pi electron clouds inside the tubulin, which is where anesthetics act here. So starting at the fast end, we have pi resonance in the terahertz region, and as we go larger, we get into gigahertz, megahertz, kilohertz, and that leaves EEG as hertz. And in our recent paper, Roger and I suggested that EEG, which is hertz basically, 0 to up to 100 or so hertz, maybe higher, as Peter Fenwick has told us, maybe several hundred or even 1,000 hertz. It could be derived from beat frequencies of much faster vibrations in the microtubules. So for example, megahertz, and these microtubules are mixed polarity, so they're going to be slightly different energy. So if they're oscillating, they're going to have slightly different energy, and like two guitars that are slightly out of tune, and we'll get beat frequencies, beats at half the difference in frequency between the two oscillators, in this case microtubules, so that we propose that EEG is actually derived from beat frequencies of faster vibrations inside neurons coming from microtubules. And I've talked about this with many EEG experts, and generally they don't like the idea, but they can't say why it's wrong. And there's no other really good explanation for EEG as one coherent system, other than saying, well, this frequency comes from this frequency comes from that. So we did propose that. So to begin to wrap up, let me say that Darwinian evolution is a pillar of modern science. I don't think anything I've said would conflict with that. The only thing that's different is that I'm saying that pleasure rather than gene survival is the feedback fitness function driving evolution. And I'm not an expert in Darwin, but I'm not sure he said that. I know Richard Dawkins and others have said that. But the notion that life evolved behavior to promote gene survival is an assumption and doesn't really make any sense for several reasons. All behavior is driven by reward. Rats in a maze, any experiment we do, even our behavior is generally driven by pleasure and avoiding pain, avoiding displeasure. And by pleasure, I include hedonism, but also altruism. It feels better to give than to receive. Spirituality, whatever feels good is what motivates us. And, you know, a lot of us have learned to delay our gratification and go to school for many years and be happier later, but that's still a form of motivated by pleasure. Also, there were no genes in the primordial soup. From the time of the primordial soup until there were genes was, I'm not sure, maybe one of the experts can tell me, millions, if not billions of years. And finally, evolutionary theory ignores consciousness and feelings. It's just kind of an ad hoc throw-in later on that doesn't really fit in. So I raised the question, did feelings drive evolution? And I've suggested a possible mechanism how this could have occurred. And this is also known, I found out recently, known as the phenomenal argument in evolution. So I'm not the only one thinking along these lines. There are people in philosophy who are questioning the gene survival idea, and that the phenomenal argument, meaning phenomenal experience, feelings, pleasure, might be important in evolution and drive evolution. Now, the last point I want to make is going back to Roger's cyclical conformal cosmology, the idea that the universe is serial and didn't just start with the Big Bang, but was preceded by another eon, and which had its own Big Bang. It should probably be small, lowercase b's maybe, which was preceded by another one, going all the way back infinitely. And this is somewhat more satisfying, I think, than saying, well, it just started so many billion years ago. And as he explained in his lecture, there's rescaling so that we see a crossover. And I should also say this evolution idea and what I'm about to say are my ideas based on Roger's ideas. He doesn't necessarily agree with them. If they're right, he should get the credit because they're based on his ideas. But if they're wrong, don't blame him, blame me. But one thing I have said in another article is that if you consider the anthropic principle and the fine-tuning of the universe question, why there's 25 physical constants that determine the behavior of the universe, the mass, ratio of the mass of the proton to the electron, the cosmological constant, the fine-tuning constant, et cetera, et cetera. If these weren't all exactly the way they are, we wouldn't have stars, we wouldn't have light, we wouldn't have life, and we wouldn't have consciousness. So the idea is that, and this has led to the anthropic principle, which is two potential solutions. I forget which is which, but one is that there was some primal cause, or the universe is doing this for some purpose or reason, and some people attribute that to God. But in this case, it would be to feel good, evolution, and because what I'm trying to say is that at these crossover points, these cosmological constants could evolve and mutate during this cycle. Lee Smolin actually presented that idea in a book about black holes and baby universes. So it could be that these crossover points, these physical constants mutate and evolve so that each eon subsequently is more conducive to life and consciousness than the previous one, and the universe itself may be evolving over eons and eons to optimize consciousness. And in some sense, consciousness is driving or steering the universe. So let me conclude by saying that according to panpsychism, Whitehead, Eastern philosophy, and Rogers' objective reduction, consciousness and feelings may have preceded life. They were here all along. If so, feelings and pleasure may have prompted the origin and evolution of life from the primordial soup to the present day. And that, and as I said earlier, that pleasurable feelings may encompass hedonism, altruism, and spirituality. So it's not just hedonism, but it could explain even our behavior coming to a conference because we enjoy the camaraderie, we enjoy learning, and we enjoy the good times we've had here. So thank you very much for your attention.
And Stuart, we enjoy you. Thank you so much. A philosophical principle is to maximize happiness and pleasure. So that would certainly fit with this idea to make it not a human construct, but a universal construct.
Right. I have to differ with my good friend Deepak, and I don't think he's here, but as he said, we've had our disagreements because he has consciousness being everything, And I think consciousness is intrinsic to the universe, and that's different from the materialist emergence ideas that consciousness emerges as a higher order effect. I think it's in the woodwork of the universe, but it's not everything.
A question in the back there? Oh, okay. Go ahead.
Stuart, you said that some people don't like your theory very much, and frankly, I don't understand this. I mean, you're not trying to sell me meaning or a massage chair. You're trying to sell me an amazing hypothesis that is, this underground quantum psi-resonance theory tries to explain emotion, consciousness, evolution, and psychedelics in one fell swoop. I think that is very, very appealing and attractive.
Thank you.
Yeah, honestly think this. Now, what's the probability that that theory is true?
Now we come to the
because and you know this problem with Roger's theory is that quantum mechanics cannot solve the Gödel problems. James can solve some of them, but not all of them. But current physics, quantum mechanics cannot do this. This is why Roger wants to have new physics to do this. And but you work within existing physics with these phi-resonant quantum underground to explain this. And it's a very interesting hypothesis. How likely do you think it is true? What's the probability that you think this theory that you made much stronger today than it was when I read up on it last time is true? And what would convince you, what kind of evidence would make you change your mind and drop it and pick a different theory?
Well, first of all, let me say that I include Roger's objective reduction, so I'm using new physics. I'm going beyond conventional quantum physics and agreeing with Roger that we need something else. As far as what would convince me or anybody else that this was true, I've said a lot of stuff here. So in toto, I don't know. But going back to the basic ideas we've been putting forth for 24 years, I think, 23 years, about quantum computations in microtubules being responsible for consciousness, I think that is on the horizon of being testable. And for example, Travis's paper talked the other day about anesthesia acting on these terahertz vibrations. So what we hope to do is utilize Honor-Bond's technology to measure and detect quantum vibrations in microtubules inside neurons, and then bring in anesthesia, and show that if you start in air or oxygen, then introduce some anesthesia, and you dampen these vibrations and they go away with anesthetic A. Then you bring in anesthetic B, which is twice as potent, and you find, for example, that you only need half as much to do the same thing. And then you wash it away and the quantum resonances come back. And you do that for a series of anesthetics, all with known potencies, and you plot the potency, how much it takes for each anesthetic to put anybody to sleep. Not only anybody, any animal, because the potency is the same for a human, for a horse, or a fruit fly. And you see if the potency of the anesthetic in dampening those quantum vibrations correlates with their ability to put you or I to sleep. So, and then you do the same thing with psychoactive drugs. You put a little psilocybin or LSD on it and see if the resonances increase, and that would be evidence in that direction. Now, that wouldn't prove objective reduction, but it would prove that quantum vibrations in microtubules would appear to be necessary, would appear to be the critical feature in consciousness. Because I think anesthesia is the best tool because it's selective. Under anesthesia, the brain's still active, and all that's really missing is consciousness.
In your theory, you're saying how life and consciousness could have evolved for pleasure, for wanting to feel good. But if we're looking at biology and nature, one of the most powerful things that we see is, even on a single cell level, is the survival instinct. If you're saying that emotion is connected with evolution, is it not also possible that the first emotion was not one of pleasure, but one of fear, one of self-preservation? And would your system still work with that in mind?
That's kind of the point Betsy keeps reminding me of. Yeah, I think it would actually. If you think of the frowny face, that could be displeasure, it could be fear, and you want to avoid that. And that could drive your behavior. So if you have a predator-prey relationship, if you feel fear, you're going to be more likely to run away or attack the other guy first. And also, if Roger's idea about non-computability, bringing in creativity, as James has been saying, if one organism has that and the other one doesn't, the one with it is going to be more clever and more likely to prevail, and therefore more likely to evolve. So I think that's fine. I just picked pleasure as one and displeasure as the other, but you could say fear or, you know, a zillion other things once you start putting them all together. Could be.
Thank you for the amazing theory. I have a question about the bacteria. So one of the forms of life is bacteria, and there's a lot of it living inside of us, on our skin, and they're constantly evolving, so they're in constant evolution. As far as I know, they don't have the mechanisms to experience pleasure or pain. So how could that fit into your theory, and also how could viruses fit into your theory? Viruses.
Well, all animal cells have microtubules. Plants have microtubules. Prokaryotes have something, simple bacteria have something very similar called the FTSZ protein. Even Archaebacterium have something similar to that. And viruses have these essentially quasicrystals of proteins which have the same pi resonance group. So I think microtubules are best suited for it, but they're not the only way to have quantum states which collapse by this mechanism. So anything that has that could have some kind of feelings and could drive evolution that way.
Stu, would you please speculate on a potential connection?
I haven't speculated enough.
Never. Between the effect of anesthetic agents in hydrophobic pockets, and then in Bruce Damer's talk, the key there being a chemical complex that is able to dehydrate itself after multiple cycles, rehydrate, and survive and expand.
Hydration is the enemy. I want to talk to Bruce about this, actually. Hydration is the enemy of quantum. Okay, so when they say the brain is too warm, wet, and noisy, or biology is too warm, wet, and noisy, well, the warm, actually, living systems, including microtubules, and also plant photosynthesis, utilize heat to pump coherent vibrations, going back to the Froelich mechanism. If you have a geometric lattice of some, and you just put energy, it's going to vibrate coherently, and that's going to help you avoid a decoherence. Also, the nonpolar region is dry. It's not wet. And I didn't quite follow what he was saying, but the hydration, if it gets too wet, that's going to hurt the quantum effect. So if you get, the drier it is, as long as it doesn't get too dry, would tend to promote quantum effects.
And just before, I just want to thank our microphone runners who had their running shoes on, Betsy Bigby and Kelly Garnell. Thank you so much for helping our people with questions.
My dear Dr. Pangloss.
Dr. Who?
Dr. Pangloss.
Pangloss.
You may remember him. What is of interest is how you might take this viewpoint and elaborate it further into explaining some of the differences, in particular, between humans and other organisms.
So Tom Bever is a language professor. He's a colleague of Chomsky, and we've been having this debate for the last year or so about language and X-bar structures, which Noam proposed and then seems to have backed off a little bit, which these are branching patterns that you can describe sentence structure with. And when I saw these branching patterns, they looked a lot like the patterns we see in microtubules. So I've been working on an idea that language may have its origin in microtubules. And I'm getting some pushback from Tom and Noam. and Massimo, our other colleague, but I'm persistent, and I'm gonna keep pushing it. So I guess my answer, Tom, would be it could be that humans have more fully developed the language potential based on X-bar structure in microtubules. So here comes his rebuttal.
Well, the question I was meant to be asking is not about that, although that's of interest, of course. It's how you could elaborate the pleasure principle to support an argument that, at least intuitively, we humans believe that certain kinds of capacities in humans have evolved that make it possible for humans to recognize certain abstract computational possibilities and so on. So how would that devolve from the pleasure principle? That's what I'm asking.
Well, first of all, Chuck Stevens' our talk yesterday was about evolution, and he showed a comparison of a lot of different brains. And in the end, I didn't really see any big difference that I could tell. Some other ideas that were put forth either by him or by others about the ratio of glia, and some other things. But what I would say is that what distinguishes us from lower animals would, assuming, because they all have microtubules, would be the MAPs, the microtubule-associated proteins, which are kind of the accessories. Tau, for example, is a microtubule-associated protein. There are many. And each tubulin, there's about 1 billion tubulins per neuron, and each tubulin can be modified. It can be post-translationally modified by taking off an amino acid or adding an amino acid. You could phosphorylate it. You could bind something to it. So each tubulin might have 20 different possibilities. So the number of information states in one neuron would be 10 to the 9th to the 20th or something like that. So it's enormous. So I would imagine, I'm pretty sure that lower animals don't have that diversity, and it's kind of like everybody has an iPhone, but a lot of people have way more apps. I just have a couple apps because I'm not big on using my iPhone, but other people can do anything. They can run the world from their iPhone. So if it's kind of like the apps are the maps, and they give the microtubules more capacity to do different things.
Hello. Here's an idea or a suggestion. Instead of using the word pleasure, may one use the word hierarchy of needs, as in Maslow. And in the Eastern philosophy, there's a 7-level hierarchy of needs. And I think that probably fits all of the lower animals as well as up to the human being, and the human being's highest need also.
Well, but just by saying, I need to feel pleasure. Because the point is that feelings drive our behavior. You know, a non-conscious entity would not know that he, she, or it needs anything. But how would it know that? Well, what's instinct? Program, but who programmed it?
Hi. So I like the pleasure over here. I like the pleasure principle. I guess 2 questions, maybe suggestions about ditching a couple of aspects of this theory that might make it better. First is the frequency issue. I disagree completely that EEG modelers haven't explained why we have certain frequencies. We explain it all the time, and it's easy, it's mostly contingent, and it's actually, has been mentioned before, a little bit of a problem rather than a solution. So this idea that frequencies, whether they're alpha or in the megahertz or whatever, are somehow the solution to consciousness, I would disagree. So I would ask, maybe we can ditch that. And the second thing I would ask about ditching is the quantum aspect. So if you're actually sort of motivating a theory of consciousness where there's a causal feature to pleasure or anything of that nature, I think that that's fully applicable to any panpsychic principle. So why not just say that that's playing a role? I don't know that the quantum explanation is actually doing much more work than just going right to any panpsychic level and leaving that question open, whether it's at the quantum level or it's at, last year I think we suggested it was the atomic level or the molecular level.
Well, first of all, the quantum state reductions, objective reductions, are events. Now, the problem I have with panpsychism is they say, well, okay, it's panpsychism. So what does that mean, though? Does that mean that qualia are painted on an atom? Are they painted on the molecule? Are they painted on the proton, the neutron? And
they're part of it, not painted.
Pardon me?
That's the point of panpsychism. It's part of it. It's not painted on.
It's part of it. Okay, but is it part of the atom?
Just like mass. Yes, like mass. It's a property.
At what level, though, and how do we differ from anything else? Then why isn't this lectern is if it's panpsychism? Why is it any different from me? And also, consciousness is a process. It's not a state, really. If you go back to Whitehead, it's a sequence of events, and I think that's becoming more and more clear. And I don't agree with what you said about frequencies either. People talk about, this rhythm comes from this pacemaker, this comes from this network, but that means that EEG as a whole is just a bunch of disconnected things. It doesn't really hang together, as far as I can see.
Do you find anesthetics modulate creativity?
You're not very creative when you're under anesthesia.
On the way in and on the way out.
It's funny. Anesthetics, they're biphasic. On the way in, there's an excitatory phase, what we used to call stage 2. So if you don't have an IV and you breathe, just have a gas, and you breathe a kid down, for example, they tend to get excited. And in fact, it was this excitatory phase that led to the discovery of anesthetics back in the 1800s, ether frolics. People were having parties by just sniffing a little ether. They had all kinds of fun. They felt very creative. And somebody mentioned nitrous oxide does the same thing, laughing gas. But if you take more of it, if you continue to breathe nitrous oxide, you go to sleep pretty much, although it takes more than one atmosphere of nitrous oxide to be a complete anesthetic. So I think at low doses, they're excitatory and might have some creativity. And then there's ketamine, which is still a mystery how that works. But that, of course, causes very strange effects, which could be considered creative. So it could be that at low concentrations, they're excitatory and possibly promoting creativity.
I wonder whether you've had a chance to think about graphene, which is a two-dimensional lattice of hexagonal rings with pi resonances.
Yeah.
And it occurs naturally in high concentration in charcoal-rich soils like Terra Preta soil in Brazil and Bolivia, which are incredibly resilient, healthy, symbiotic ecosystems of mycorrhizal fungi and bacteria that seem to be gardens of many levels of satisfying needs for the microbiology of the soil systems.
Yeah, graphene is like a two-dimensional sheet of benzene, so there's a lot of pi resonance there. So yeah, I think that if I would consider what we call the quantum channel or the quantum underground spiraling through microtubules to kind of be a ribbon of graphene, except that the graphene they're actually connected. So here there's a little space so they can oscillate back and forth. I don't know if that oscillation occurs in graphene or not, but they're very similar.
Stuart, thank you for that. I was surprised that you didn't refer to one point, which is it seems to me that under a mainstream physics and neuroscience explanation of consciousness, it can't possibly have evolved because it has no active influence in the universe. And isn't that, if I were arguing your case, I would first say, this is the fact that we are inside experience is the single greatest data point against the mainstream science case.
Yeah.
Make that point first, and then elaborate it, perhaps, as you do. So perhaps you could comment on that.
Yeah, thank you for that. There are many articles. There was one recently in a cover article in New Scientist, Consciousness, what is it for? What good is it? Because the idea that it emerged at some point, and now it's just along for the ride, and that goes hand in hand with the idea that it's epiphenomenal. And as T.H. Huxley said, summarizing that viewpoint, we are merely helpless spectators, conscious automata along for the ride. And therefore, free will is impossible, or there would be no need for it. I guess you could argue that it then took over. But yeah, I think this certainly gives a purpose to consciousness, and not only in terms of driving evolution, and really biology is a vehicle for consciousness rather than consciousness being along for the ride. You just turn that around.
Okay.
Very insightful talk. So question is that what kind of diet do you follow? And can you change your consciousness with your diet? If everything what you said, it should work that way.
I couldn't hear what Betsy said, but she's always trying to get me to eat healthier. And when I do feel better. So, well, tryptophan, but too much tryptophan, like in turkey, makes you sleepy. So I don't, just eat healthy. And I eat a lot of fish and oils, fish oil, salmon and whatnot, which has a lot of that kind of stuff. So I can't answer that. Actually, Betsy could probably answer it better than I could.
And with that, we'll say thank you so much, Sue.
Allow me, please, Eric, but don't leave. I want to thank, first of all, Abby. Is she here? I think she just left. But she's absolutely amazing. This whole changeover from Shanghai to here was a mission impossible. And we did it thanks to Abby, Betsy, James, and Eric, and Kelly. And we got some flack from my university because they go, what the hell are you doing? And they thought it was a big liability, but we fought through. And the hotel. So, and I want to thank the program committee, James, Eric, Jay, Pavo Pilkinen, Betsy Bigby, my fiance. And I want to thank, and also the mic runner, along with Kelly. UCSD for receiving us and cooperating and giving us a bunch of really great speakers. Thank you, guys. We didn't have to pay travel, so we saved some bucks there, and because it cost us actually moving over. I want to thank our AV guys. You guys are freaking awesome. I can tell you, having run a lot of conferences, that AV is essential, and these guys are wonderful. We brought them from Tucson, right? Are you guys? Yeah, we brought them from Tucson because we worked with them for years, and they're wonderful. Commotion Studios, right? Betsy, the mic runner, Kelly, the registrants, you guys who paid to get in, you made this possible. Thank you so much. Our sponsors, our excellent speakers. The talks will be online in about 3 weeks if you go to our Center for Consciousness Studies. Is that Abby? Abby, I just thank you. Let's give her a round of applause. Amazing, amazing Abby. Nick Day and the Consciousness Central team, and they've been putting out broadcasts on the web, and if you go to our website, you can see them. And unless I've forgotten something, I just want to end by saying there's a party tonight, the last party. It's going to be right here, about 8 o'clock. There will be food. There will be drinks. You'll get a couple tickets for free drinks. I, for one, plan on drinking heavily tonight. And it'll be low-key. We're going to have some music. They're going to provide piped-in music. Dorian's packed up, and let's thank Dorian Electra and the Electrodes and Qualiatic, who were sensational. There she is. All right. And we still have her jingles in our. What was that one, Betsy? That was in your head? Huh?
Don't talk about earworms.
Brain in a Vat has been an earworm in Betsy's head since last night, so thanks a lot for that. What was that line? Big hair and endless croissants? Is that a... Yeah. All right. So let's thank Dorian Electra and the Electrodes, Nick Day and the Consciousness Central team, all the speakers, all the participants, all the helpers, the hotel, everybody. And thank you very much. See you tonight.